I don't know. You guys. So when we're talking about gel electrophoresis, we're talking about seeing the really tiny, the molecules. It's about seeing molecules. That's intense. We're talking about things that are a few hundred atoms or 1,000 atoms long, 20, 30 atoms wide. Like it's how do you see that? Like you can't see it with your eyes. We have some electron microscope work we can do to see really tiny things, and in fact I have Rosalind Franklin's. We have a really great case study. I don't think we have enough time to do, unfortunately, before the test, but it really is a fantastic case study that takes a look at what we saw when we first figured out this shape that you guys are all so familiar with. It's on your school logo, the shape of DNA. I know you've heard of Watson and Crick. So Watson and Crick depended on Rosalind Franklin. Nobody knew for sure this shape of this molecule because we can't see it. We knew DNA existed. We knew DNA existed before we even knew what it did. You took some blood, you took some skin, you took some hair, you took an eyeball, took a liver, you chopped it up, you looked at it. What did you see? You see pudding. Like, what do you really, how do you get to see these molecules? Well, what is it we can do? Rosalind did something amazing. She did not get the Nobel Prize because she had died before the Nobel Prize came out. It's a really big scandal because it was a misogynistic culture and she was treated pretty badly. She wasn't accepted by her male counterparts, and Watson and Crick stole her work, actually, to be fair. Did I tell you this? I'm good. I just keep saying it because people need to remember that. Like, everybody remembers Watson and Crick, but nobody remembers that what happened there was, I don't know who did it, but Watson and Crick were the beneficiaries of stolen information. Yeah. Say it. Yeah, her coworker she was working in a very male chauvinist lab, misogynistic lab. Her coworker was under the impression she was going to be his assistant. She had a PhD. Her specialty was making crystals of organic molecules like proteins and DNA. It's very hard to do a crystalline structure. He was very resentful of her. Her life there was not easy, and she had decided to move to another lab. She couldn't keep her work because the lab owns the work. Whatever you do, that organization owns the work that you do in there because they're supplying you with money and supplies and such. Before she had left, her coworker went to her desk and stole the picture of DNA that she took. And then her department chair had something to do with it as well. Ended up giving it and showing it to Watson and Crick, and they figured out the shape of DNA. And because they figured out the shape, they can then figure out how it worked. So we couldn't have figured out how it worked unless we had some idea of what we were talking about. Because we can't go from pudding to transcription and translation. You can't do that, right? Like, how do we learn transcription and translation, right? There it is on the board. How do we know that those are the steps that actually occurred? Many, many, many years and many, many, many thousands of experiments and hundreds of people to figure out it took you, well, some of you still haven't learned it, but what took you, a couple months to learn. So what you're the beneficiary of is years of work, and it's been distilled for you to learn quickly. And that's good. But sometimes we forget that, and we kind of blow off how simple things seem. It's not that simple. So Franklin's picture was a fuzzy thing that looked like that. And from this picture and some other information, Watson and Crick were able to come up with this three-dimensional shape, and then later the function of DNA. This fuzzy thing that looks literally a lot like this. How did they do that? They understood physics. They understood how this is called X-ray crystallography. And you know me and spelling. So X-ray crystallography is just taking X-rays of a crystal that you've made, and you can make crystals. And crystals are very, very stable forms of a molecule. You can't take an X-ray picture of a molecule that's wiggling. So a crystal is just a very still version of it. The trick is to make a crystal of a molecule in its natural shape. You can make crystals on anything, you can freeze it, but will it be in the shape that it's normally in? That's the question. And Rosalind was able to do that after many, many years and hard work. So anyways, the point is, how do we get these, that's just one example of how we looked at these tiny little structures called molecules, and we can understand what they are. So how do we separate them? We got this pudding, we got this soup. We take your liver, we throw it in a blender, literally, that's what we do. Throw it in a blender, we mash it all up with a perfect solution, so it's a buffer solution, so it's not too acidic, not too basic. Mash it all up. Now we got this soup of molecules. In that soup we have RNA, we have DNA, we have, somebody close that door. And many different kinds of RNA, right? And at least in a human, at least 46 chromosomes worth of DNA, not to mention the DNA in ribosomes. Then you have, not in ribosomes, in mitochondria. Then you have lipids, many different kinds of lipids, including phospholipids, cholesterol. Then you have carbohydrates. And then you have proteins. And again, hundreds of thousands of different versions of each of these. Insulin. Close that computer. Insulin. Glucagon. Testosterone. Estrogen. Acetylcholine. I mean, the list goes on. A whole book full of molecules, all in this soup. How do we isolate any one of these? Well, the first step is to use chemistry. We have a soup, and so one of the things we could do is we could use the fact that some things float and some things don't. Some things are fat soluble, some things are not. Some things are polar, some things are not. Like dissolves like. So we can put in a non-polar solution or in a polar solution, and we can start separating things, right? So then we have these jars. We have these jars of different solutions. And in each one, using that chemistry, we're able to separate them into RNA, DNA, lipids, carbohydrates, and proteins. You know, the interesting thing is that one of the easiest ways to separate lipids is actually to use a paper bag. Because do you ever see a grease stain on a paper bag? So paper bags the lipids will stick to the paper bags. That's kind of interesting. But there's all kinds of chemistry. When you take chemistry, you'll learn one of the big things about chemistry is separating substances and then identifying what those substances are. So chemistry can get us this far, but biology, something we call, yes, bless you, something we call biochemistry, which is a big part of biology. And of course I'm going to misspell chemistry. So a big part of biology is biochemistry. Is how do we separate then the individual RNAs? How do we separate the individual DNAs? How do we separate the individual proteins? That's really and even the DNA. How do we know? Like you've seen this DNA sequences, right? You've seen them in problems. How do we know it's A, G, C, C, T, T, A, T? How do we get to that point? We have to continue to separate. Separate. Now we're talking about separating not just molecules from each other, because here this is kind of easy. We know, for instance, that DNA and RNA have phosphorus in them. We know lipids are nonpolar. We know carbohydrates have carbon, hydrogen, and oxygen. And we know proteins can be polar, nonpolar, but have nitrogen, sulfur, right? So because of that, we can separate those. We can put them in a tube that attracts sulfur, for instance, and you're pretty sure you're going to get nothing but proteins, right? And if you put it in a non-polar solution, you're probably getting mostly lipids, that kind of thing. But how do you separate the individual pieces and parts that are inside the components of that soup, that original soup that had everything in it? So we went from this soup to use some chemistry to separate. Then now we're going to use biochemistry and individually separate the components of, let's say, DNA. So you have this long molecule of DNA. You have two of them, two samples. We've separated them using chemistry, using some biochemistry, but mostly chemistry. And there's sample 1 and sample 2. The example people always love is there's a murder mystery and we have two individuals and we want to know, is this DNA, one of these DNA samples were found at the murder scene. We have two suspects. Which one did it? Well, we can use these things called restriction endonucleases. Now, those are enzymes that we find in bacteria, specific bacteria that live in really hot springs. And in any case, bacteria have these, restriction nucleases are found in all bacteria. Sorry, excuse me. I was thinking PCR. But restriction nucleases, bacteria have them because bacteria have to destroy viruses. Viruses attack them all the time. And their immune system is not as complex as ours, obviously, because they're only a single cell. So they need something that's going to help them fight off viruses that are made of DNA. And DNA, they only have one chromosome and a bunch of plasmids, so how do they make sure that they don't get some foreign DNA in them that infect them? So they have, yeah, you can go and do what you, just take the pass. So they have these restriction nucleases that cut DNA. They cut DNA in specific sequences, in specific places. And of course, the DNA that these that are inside, let's say E. coli, Eco R1, Eco R1, that's a strain of bacteria. This strain will have a restriction on nuclease that cuts DNA. I'm making this up, but let's say it cuts DNA at ATGCTTA. If it sees this sequence, it's going to cut it. Like molecular scissors. Every time it sees this, it's going to cut it. Do you think it has this sequence in its own DNA? No. So every species of bacteria that has these restriction of nucleases, they have restriction of nucleases that cut DNA that's not in their genome. All right. But that's easy for them because they have a small genome, right? It's one chromosome. So, it's a big one, but it's still one. And then small plasmids. They don't have 46 chromosomes, right? With 2 copies each from 2 different parents. Like that's a lot of variation. It's likely that this sequence does exist somewhere, multiple times, somewhere in this DNA, right? And these are 2 different people, correct? Yeah. So this EcoR1 restriction of nuclease will cut the DNA in different places in different people. So yeah, it's going to cut so if it cuts it in different people, what you're going to end up with, and I'm going to use, let's use green for this one and red for that one. The same restriction nuclease is going to cut this sequence, and it's going to cut it in different people in different places. So in number 1, you're going to get a piece of DNA that's this small, and you're going to get another one that's about as small, and then you're going to have one big piece. That's what you're going to get. And over here, you might get 4 pieces where of varying lengths. I should make this red so we can see that it's from 2 different people. Now we have the same chromosomes, but because the sequences are different, the DNA is going to be cut differently, and so you get 2 different sizes. Here you only have 3, and here you have 4. Here there's 2 that are same size, 1 big one, and 1 medium-sized one. Here's 1 big long one and 2 little ones. Can we see this when it happens? So you grow the bacteria, you isolate the protein because we can use chemistry to do that. We got the restriction of nucleases. We know it cuts ATGCTTA. We throw the enzyme in with the DNA from suspect 1. We throw it in the test tube with suspect 2. Do we see the pieces? Can you hold the test tube up to the light and say, "Ooh, this one's this and this one"? No. How do we see it? Gel electrophoresis. That is what's hard. That's why people spend years. It's a lot of time, but we figured it out. And actually, technology's advanced. It's much quicker now. When I was doing this, I was using radioactive isotopes. When I was sequencing DNA, it was radioactive isotopes and cloning them in bacteria, wearing iron aprons so I wouldn't get, lead-lined aprons so I didn't get exposure to radiation, etc. All right, anyways, so we're going to take advantage of the fact that when you make this thing, there's this thing called agarose. That's just one type. We used to use polyacrylamide. But in any case, what we're going to do is we're going to make a filter. And agarose is a seaweed. Man, I'm just all over the place this morning. Seaweed protein. So it would make, you eat Jello gelatin, correct? You've had it before. That's made from pigs, protein from the pig's bone marrow. So that's why Muslims have never had Jello gelatin, at least not from regular Jello gelatin or Jewish people. But there are gels that are made from seaweed and other proteins that basically the same thing. So we use agarose in our cooking as well. So you can make something like Jello gelatin. Yeah, somewhere. If you watch Chopped, thank you. If you watch Chopped, you'll see them use agarose to thicken soups and things like that. But you can actually make gel out of it. It's not as wobbly as Jello gelatin is, but it's still gel-like. Yeah. Huh? Ah, thank you. Yes, we used it. That's right. I forgot. We did it during our osmosis gel. That was agarose. That's a gel. And we're going to do it again. We're actually going to do one of these. But I don't feel like I want to spend the time before the test doing it. So we'll do it after the test, okay? So we're going to do it. We have the equipment. We'll make the gels. So we're gonna play around with this stuff, but after the test, okay? So agarose, what we do is we take these proteins and they stick to each other really well, randomly. They stick to each other. This is kind of a cross section of it, and what you get is they form this matrix. If you can imagine that you have this kind of a filter. You've seen filters before, screens, yeah. All right? But really, really tiny screens, really tiny holes. So that what you end up with when you have it is this little sack. You've seen it. We had little cubes of it. And it's between two glass plates or plastic plates. I'm not going to draw that. And what we do is we put what's called a comb. Oops, that's way too big. I don't know why this does that. So we put a comb in so that we can make these things called wells. When I mean comb, I mean we could literally put something that looks like a comb with teeth. And then when the gel forms, when it turns into the gel and goes from liquid to a gel, it leaves these little wells. The wells are just spaces that we can use to put samples in. We're going to put samples in there. The samples we took from our prisoners, for instance. Well, not prisoners, but suspects. I already convicted them. The mock trial kids would be very upset with me.
Yeah, the mock trial people, they're somewhere.
Sorry, I'm trying to do this quickly and I'm messing up these lines, but you get the point, right? So we got these wells, and then we have to have a way to make the proteins move, right? So we know we can use this thing called electricity, right? We can say, put a negative anode here and the cathode down here, or you can reverse it, whatever, but you can do something like that. And then you put the whole thing in another tub, in a bath.
Isn't it full of salt water?
And salt water, yeah, basically. It's a buffer. So the acidity is constant, and the salt. So weak acid and salt solution. And then you take pipetmen that we have in the back. They look like this, little tip. You've seen them in movies and such. As a little window, numbers. And you're going to put a specific amount of the sample of DNA that you've isolated from, let's say, from 1 here and from person 2 here. And they're going to be filled in. They're going to be full of dye. So the dye, there's going to be color to them, but the color comes from what you added to it. Does that make sense? So you stick to this DNA, this dye that there's the color is not normally there. It wouldn't have any color, but we added color to it. And then usually you have this a marker, what we call a standard. You need a control group, right? Any good experiment has a control group. Your control group is your standard. You're gonna use that to compare it to the other two. Now, there's 2 ways that electrophoresis can separate particles. We can separate them by size, depending on how you do it, how you prepare the sample, and what it is you're separating. The mechanism stays the same, but you can either separate it by size or you can separate it by charge. You can do either one. So the more negative, in the case of charge, the more negative the molecules are, if they're all the same size, the more negative they are, the faster they move down here. The more positive they are, the more likely they are to stay around here. Does that make sense? Because this side's negative, this side's positive. So opposites attract. So the more negative it is, the further. Now, if they're all negative, they all have basically the same charge, then they're going to separate by size. The bigger they are, the slower they move. Pretty straightforward. The smaller they are, the faster they move. So when we look at our standard, I'll make our standard red. You know what? Since we already used red, let's take our standard yellow. So we'll have, after we've run it for a while, we know this is, I don't know, I'm making this up, 100 base pairs. What do I mean by base pairs? Let's say 1,000 base pairs. DNA, that's right. A is base paired with T. G is base paired with C with G, T with A with T. So if it's cut at one place and it's 1,000 base pairs, a big piece is going to be here at 1,000, and maybe down here it's at 50 base pairs. I don't know, I'm making this up. So 50 base pairs down there, 1,000 base pairs up there. That gives us a standard. We know what's in this one, right? Because it's our control group, correct? Yep. We know the size and what we put in there, so we know what it should look like. So theoretically, if there's a band here, we know that's at 1,000, right? If there's a band here, we know that's at 50. Now, on your end of course exam, you're unlikely to get this kind of detail. What you're probably going to get is bands that look like this.
Oh yeah, they did have that.
And maybe one band will be really close to the other one, or maybe there'll be one big thick band, and then one that's a little. I'm sorry, I misdrew that. I'm trying to look at what we did on the. There'll be one band there, and then there'll be one big thick band. Either two bands really close together, one big thick one. Hard to tell when you're doing these sometimes. Depends out how long you let it run, how big the gel is. If it's a really long gel, there were some gels that were 2 stories high, and they would run it down. That gel, well, not 2 stories, it's too big, but very long gels. The longer you run it, the more separation you can get. Then maybe you have one really big one. One band up here, and then another one here, and then you have 2 over here. So And then of course you have the DNA found at the crime scene. And so now so this is what the DNA from person two looks like. This is the DNA from person one. Person one had two little ones and a big one, two little ones and a big one. Person two had one really big one bigger one, and two medium-sized one big one, really big one big one, and two medium-sized one. So person one had 3 bands, person two had 4 bands. Now you have the murderer, the person who did it, whose DNA is found at the crime scene anyways, whether they did or not, who knows. And then what you see is this. 3 bands in that position. Who did it? Whose DNA is it? It's certainly not person 2. It's person 1. See, they match up?
Oh, yeah.
They match up horizontally. This is the unknown DNA, it's right there. This is the unknown DNA, it's right there. So they line up. These are the 2 that are the closest together. Does that make sense? This is obviously someone who's this person, assuming we use the same restriction, everything else is kept the same, then we know that this unknown is likely to be related to person 1. Does that make sense?
Yeah.
Now we can do much more specific gels, but this is a quick and dirty comparison that we can do. This is much more accurate than even fingerprints. So if we see, that's why we call it DNA fingerprinting. We could sequence the DNA and get real detailed and get almost 100% on it, 100% match or negation of which DNA matches the DNA found at the crime scene. All right, so we could do the same process with proteins. We can do the same process. I mean, obviously, we're going to have to change things up. You're not going to use restriction on nucleases. But we can use the same process with proteins, the same process with RNA. So there's no reason for us to... This is how we see molecules. This is how we know what molecules are where. With something like this. There are many other tools at our disposal, but this is a big one. Gel electrophoresis. So we only have 3 minutes, but I'm hoping that we could go look quickly at a gel electrophoresis question that you guys took on the benchmark test. And it was probably in the genetics part.
Most likely, yeah, because there was one where it was like completely DNA, which one is similar.
All right, so I'm just going to pick an individual. Let's just use, I don't think Mayar will mind if we use hers. Let's go right to heredity. Let's see if we can find. Here's one. So gel electrophoresis, there's 1, 2, 3, 4, 5 species. Oh. Sorry. I'm going to keep the tape recording so that the people at home, or the people that see this at home, we're looking at question 46 on Mayar's test. I don't know if they're different numbers for you guys. You see there's different species of I think these look like mussels. These are different DNA samples from different mussels. Mussels are like clams. You see you have a marker. Here's your control group. Your marker is what we know that this is 5,090 base pairs. This is 2,036 base pairs. And we're cutting them up in a known way. So this is what you would get, right? If you cut up with this particular restriction of nuclease. This is what we'd expect of these different sizes. And each species is going to get cut in a different way, right? So here's this person, this Pariaceae. Now the question is, select 2 pairs of species that share the greatest genetic similarity based on the results of gel electrophoresis. So what am I looking for when I look at all these lines?
You're looking at they end up on the same, they're similar in size, and they're like on the same like line area.
That's right. So the same pattern. A lot of this is just pattern recognition. So here's two little ones and one down here. I think this is pretty by itself. You agree? These two are identical,
Yeah.
right? The pattern that we see, where these two are very close, but this has an extra band. So these two are likely the most closely related. Do you agree?
Yeah.
I mean, she chose two of them.
You have to choose two pairs.
Oh, okay. So then these two and then these two.
At the top.
Yeah,
Yeah.
I chose that.
So this one and this one,
Yeah.
and then this one, this one.
Yep.
Are we good? All right. That's all there is to it. So gel electrophoresis is very simple. It's just you have to contextualize your knowledge. Did that help?
Yeah.
All right.